let's continue our discussion of photosynthesis. I'm talking about the first step here, which is the electron transport chain of photosynthesis. You should see some similarities to what we saw in cellular respiration. Go ahead and take a look back at your notes if you need to. Go ahead and pause it. Look back at cellular respiration. And kind of look at the electron transport chain we saw. Of course, one of the main differences is in cellular respiration, kind of the last thing to occur is that in the electronic process. A lot of things that we see in here are reversed from what you'll see in cellular respiration. For instance, if you'll remember, O2, oxygen, is the final electron acceptor, the final step in the chain. Here in photosynthesis, we have water being split into O2 H plus ions, which is the reverse again, right? Again, the water was being formed in this direction in cellular respiration. Here it's being split. Kind of the first thing. Plants need to uptake water. This is one of the reasons why. So it's split, drops off two H plus ions and electrons, and light comes in and basically excites these electrons in this electron transport chain, bouncing them from higher to lower energy states across these proteins. And as it does so, it does, much like cellular respiration, pump H plus ions. It pumps H plus ions from the stroma into the thylakoid space, right, or the lumen. So it's going to pump H plus ions into the lumen right here, in the middle of these little thylakoid stacks. And so, as you might guess, you're going to end up with a high concentration of H plus inside the lumen here, which naturally it is going to want to go outside. It goes outside through a molecule you've seen before in cellular respiration. Must be a highly conserved type of protein, and it must be very important, and it is for the purposes of the test too. In fact, it's the only protein that is required to kind of understand. And that is ATP synthase. You should see some roots in there. ATP meaning this is ATP. Synth meaning it's synthesizing. There's the synthesis of ATP. And ACE kind of indicating that this is an enzyme for the process. So, again, we have H plus ions being pumped due to this light energy exciting electrons in these chlorophyll molecules that are embedded inside these proteins. Electrons are kind of being passed down this chain. Of course, it is important to distinguish between these two. This is played on here, but this is photosystem 2. And over here we have photosystem 1. These are the chlorophyll-containing proteins that are going to help to move this process along. Or again, you should remember chlorophyll. Chlorophyll is a very important molecule. This is what light is actually exciting. As it kind of strikes, as these photons kind of strike the chlorophyll molecule, they excite electrons to pass their way through this whole system. As again, H plus ions, high concentration on the inside of the lumen. The ultimate result of this process is they're kind of being forced through this very tiny opening in ATP synthase with an amount of pressure that's causing this ATP synthase to kind of rotate and slap phosphate ions onto ADPs. These inorganic phosphates get slapped onto ADPs to form ATP. This ATP that is generated, around 18 ATP or so, I believe, goes off to the Calvin cycle. Another thing that is generated is something called NADPH. You'll remember those from cell respiration, NADH and FADH2. These are electron carriers. They carry energy for us. Just to remind you, we have those two molecules. This is a very similar molecule, NADPH. The difference in this process is that we are not using these electrons for the electron transport chain. We're actually attaching, this is our electron acceptor. It's kind of acting like oxygen and accepting these electrons. And we're taking that stored kind of energy and those stored electrons over to the Calvin cycle. So ATP is going to the Calvin cycle and NADPH is going to the Calvin cycle. And as you'll know, it kind of looks probably somewhat similar to the cycle that we saw in cell respiration. Go ahead and pause the video and remind yourself what is that cycle called. All right, should have said the Krebs or citric acid cycle. Those are two names for the cycle in which carbon is kind of stripped off of glucose in very small stepwise enzyme-mediated process, and energy is generated in the form of ATP. This process is kind of the reverse. We have CO2 kind of entering, right, instead of those pyruvate molecules. We've got a gas entering the process. And we have this enzyme here, rubisco. It's the most abundant enzyme on the planet. Most abundant enzyme on the planet. And what this protein aids in, or this enzyme aids in, is it attaches these carbons from this carbon dioxide onto these molecules. It attaches onto this ribulose biphosphate, which is over here, a 5-carbon molecule, and it attaches them. There's 3 of them, right? 3 5-carbon molecules, and then it attaches these 3 carbons from CO2 onto the end here. So we have 3 added to the end. These are then broken down. We've used some ATP and ADP in this process. We used that NADPH electrons in this process to form G3P, which is a 3-carbon molecule, which is going to go out of this cycle and make a glucose molecule through some sort of reverse glycolysis-like process. So G3P, one of them exits the cycle. Of course, we have 5 left over. These 5 G3P go through this cycle again. More ATP is used in order to regenerate the cycle, right? This is a pretty important process for step because the cycle's got to occur again, right? So really only through one turn of this cycle, you only make 1 3-carbon molecule. So in order to make a glucose molecule, how many turns of the cycle would you need? Go ahead and pause the video if you need to think about that. All right, so if you said 2 turns of the cycle, you'd be correct. First, you have a 3-carbon molecule and G3P here. And that's going to go on to become glucose. We need 2 of those. And the rest of these G3P, again, are recycled in this process. You have 5 3-carbon molecules. You end up with 3 5-carbon ribulose biphosphates up here. And the cycle begins. Just go plant these carbons on here. And this is how that process works. So, in summary, photosynthesis generates ATP. It generates NADPH. Electron transfer chain generates these 2 things. They come over to the Calvin cycle, and sugar is generated. Calvin cycle. Another interesting note: the Calvin cycle used to be called the dark reaction. We don't think of them as that anymore. We also now call them the light independent reaction. This is another name for the Calvin cycle out here. Recognize, because that's another thing is referred to as the light independent reaction. I guess why it occurs independent of light. The previous cycle we talked about, these, as you might guess, light dependent. Right? These do depend on light. So this is just another way to think about the entire thing. And by the end of it, of course, we have some glucose molecules, and we're ready for those to be eaten by some heterotroph and provide them with energy. Of course, without this constant input of light, this whole system breaks down. So keep in mind, look at the big picture. Think about that next time you think about the plants outside, the plants that are being cut down or burned down in areas of the world. Think about how important these photosynthesizers are to us in the long term. All right, that's it for my video. That's all I'm going to talk about. Thank you.